Okay, so scientific inquiry, we are on page 2 of the notes packet. Um, Scientific inquiry is a universal approach to scientific problems that is similar in all areas of science. Right here, the word universal basically means that it's something that everybody can do. So scientific inquiry is pretty much about learning to solve a problem in a specific way where you end up with the best kind of answer. And that's what science is all about. So to go through the steps of scientific inquiry, my bad, let me just put up the screen shade here. Right, science is a process, first of all. A process means that science takes time in order to happen. It's not something that's going to happen immediately. When I was in the lab, it took about 10 years to be able to come up with something that could come over to the market in terms of medicine. So it's something that does take a long time and it has steps, right? So the steps that we usually take, first you have to define your problem or ask a question. Then you research your information. You create a hypothesis or a prediction of whatever it is that you want to answer. I'm going a little fast because I know that we've covered this in class already. You design a controlled experiment, which has two groups, usually a control and experimental group, and you collect data or make a graph. The reason that we make a graph is so that we can actually interpret the data a lot easier. You form a conclusion and then you repeat or peer review. Remember that when you do peer review, someone is checking it who is an equal, and that way they can tell you whether the experiment was done in the correct way or not. Moving on to the next page here, right? Information collected using the senses and tools are known as observations, and inferences are conclusions based on observations. Once again, I'm moving forward quickly because I know that we've covered this in class already. Okay, a hypothesis, right? A good hypothesis is a logical prediction to a question. A hypothesis must be testable and should never be written as a question. Very important. You never write your hypothesis as a question. Okay, experiments cannot prove a hypothesis. They can only support or fail to support it. So like I said before in class, your hypothesis, it's never going to be wrong or right. It's just that for this time that you tested it, did you prove yourself right or did you prove yourself wrong? So if you proved yourself wrong, what can you do next time to kind of get the result that you want, or do you have to revise something about your experiment to make it a little bit more accurate? That's what you're doing. Okay. And then we had done this example in class, so I'm not going to repeat it, but I just wanted to at least scroll through it in case you missed anything. Um, so we have the information right here. Once again, I'm just scrolling through it. If you want to pause or if you missed anything, we'll have it there. The results, by the way, for each class is slightly different. So if this doesn't look exactly like what you have in your paper, it's just because it was from a different class and they gave different answers, but it's about as similar as it's going to get. Okay, from this point forward, this is new information. Will you design an experiment? Okay, so an experiment is a series of trials or tests that are done to support or refute or disprove a hypothesis. So what that means is that we basically do experiments in order to see if our hypothesis is correct or not, right? We're basically trying to prove ourselves right or wrong and then move forward with what we're trying to learn or what we're trying to investigate. So that's what the purpose of a hypothesis is, and the experiment, right? You have the independent variable. So in the experiment, the independent variable is what I control. Independent variable, this is what I control. Okay? And the dependent variable, these are the results. So this is what do I want to see, right? What do I expect? And what do I want to see? Okay, so this is like basically what I'm trying to observe, the dependent variable. And this is the data that I'm collecting. Whatever information you're collecting is the dependent variable. Again, this is usually some kind of number or figure that's dependent. Something that you're going to see, a change, right? Now, in a controlled experiment, you test only one independent variable at a time by using two groups. So again, you're only testing one thing at a time. If you test more than one variable, then the experiment is not going to be valid. You don't really know what caused the change if you have too many things affecting the experiment. So you have to test only one single variable at a time. And for that, you have your control group and your experimental group. So your control group is the group that's going to be under normal conditions. And the experimental group is what's going to be changed or different. And this is what's going to receive the treatment or the independent variable. Okay, so you have those two groups at least. Now, with the experimental groups, you can have more than one group. You can have like 10 experimental groups or 5 of them and still one control group because you have to compare it all back to the control. Okay. Now, for control versus experimental, once again, what receives the unique treatment is going to be the experimental group, right? This is what's going to be different. And the control. The control is going to be exactly the same as the experimental group, but it's not going to have the independent variable. So that's the one thing that's going to be different. And this serves as a basis for comparison. So you can compare whatever you experimented on to whatever seems normal. Now, with humans, we have to use something called a placebo. Okay, so this is a little bit different, right? So with humans, if you're testing, let's say, a medicine, you can't give one group the medicine and then not give it to the other group because the humans will know, oh, I'm the control group because I didn't get anything, and they can talk to the other people in the group. So in order for humans to not know what group they're in, so that they don't know that they're really part of the experiment or if they're part of the control, you give them a placebo, which is a fake pill, right? A placebo is like a fake pill. And that way, if they take a fake pill, they think they're part of the experiment, but they're really not. It might be a sugar pill. It might be something that doesn't really affect them. And so again, with human subjects, you might use a placebo in order to experiment on them because you don't want them to know that they're actually receiving a certain treatment. Okay, now, again, a placebo is a treatment that has no effect, which is administered or given so that all groups experience the same conditions. Remember that no group should know whether they're the control or experimental. The placebo effect, this is also very interesting. So in a placebo, believe it or not, people in the control group actually report improvement. So when humans show a response to a placebo or a fake treatment, about 20% of people actually given a placebo will report improvement. And placebo results are not valid data. So this has to do with psychology. So you gave these people this fake pill, and they're saying that it made them better. 20% of people will do this. So what that proves to you is that about 20% of people are psychologically like ill. You know that they have a disease, but it's really in their head whether they get better or not because they're responding to something that isn't even real. So that's the placebo effect. Moving forward from there. Are constants, right? We talked about this in class, things that are going to stay the same, right? The keyword here is the same or consistent keyword. Now, all factors should be constant except the independent variable. So that's the only thing that's going to be different. And then some examples of constants, right? Let's look at this picture. So we have plant 1, 2, and 3, right? This plant has no water. This one's being watered every day, and this one's being watered every day. This one's getting 20 milliliters, this one's getting 40, and this one's getting no water. So that, when I'm coloring in blue, is independent because that's what's different. But what I'm going to circle in green is what's staying the same. The type of plant is staying the same. The sunlight is staying the same. Probably the amount of sunlight is staying the same. So we're keeping everything the same except that one thing. So these are the constants, the things that we're keeping the same. Right? So some factors that you want to keep constant. I'm going to just cover some of them because I want to go through them one at a time. Right? When you have humans or animals, you want to try to have the animals be as similar as possible. So humans are all different, but how do we test let's say a group that is as similar as possible. Well, you would try to have them have a similar diet, try to find people of similar fitness level or exercise level, people that are similar age and gender. So for example, if I'm doing a test, let me test only the females between ages 18 and 24, something that they have in common, right? People that weigh around 150 pounds or whatever, you have to be specific. You want to try to get people that look similar. Sleep cycle, people that sleep in a similar cycle, and then people that are similar weight. Now with plants, what kinds of things do you keep constant when you're doing an experiment? You want to keep a similar amount of sunlight, similar amount of water, similar type of soil or pH of the soil, the acidity of the soil, the type of plant, and the size of the pot. Okay, now, when you collect data, okay, collection of data and analysis. So data means that you're collecting information, basically. So data and observations can be compared between groups to make inferences and draw conclusions. So pretty much you're collecting information so that you can go ahead and analyze it and say something about the information that jumps out at you. That's what you're doing this for in the experiment. Your conclusion should compare how your results related to your original hypothesis. So in your conclusion, you're gonna say, my hypothesis was refuted or my hypothesis was supported. So yes, it seems like I have the right data, or no, it seems like the data did not go in my way, so I'm going to change my hypothesis. Now, you're going to say whether your hypothesis was validated or not, supported or refuted. Again, were you closer, or are you getting farther away? Are you getting warmer, supported, or are you getting colder with refuted? It says results should be consistent and repeatable in order to be valid, in order to validate one's work. What that means is that someone should be able to repeat this experiment for many years and get the same results in order to say, okay, this is valid, this is true. It has to happen for years, okay? Now, if your work is repeated and with similar results over and over again, your data is considered more reliable. So the more you do the experiment, the more reliable the information tends to be, or the more believable your experiment seems to be. If you only do something once, people can't really believe you or trust you because you haven't proven it many, many times. Now, improvements for future experiments, right? So one thing you want to do to make your experiment better, you can always do something called increasing the sample size. So for example, instead of using 2 mice for a study, we're using 30 mice, right? Large experimental groups result in more validity. So when you have more people, more animals, more things that you've tested, it makes you seem more believable. That's what we're talking about with validity. You're more believable. Another way to improve an experiment is to repeat it, right? Do it over and over again. And the last thing is peer review. Peer review is when scientists evaluate each other. So again, if someone's evaluating you and they're also a scientist, they know exactly what to look for in an experiment that's going to make it successful. And that's why you want to have a scientist peer review. So again, 3 ways to improve the experiment: increase the sample size, repeat the experiment, and do peer review. Okay, last but not least, let's go through the difference between a law and a theory. Okay, let's do shade here, right? So a law, a fact about how the universe is. It can be used for prediction because laws are based on mathematical formulas. They are completely true. So for example, gravity, right? Gravity is something that we can measure with a math formula. 1 plus 1 is always going to be 2. It's not going to equal 5. It's not going to equal 6. And this is how the universe works, laws. So there's all these laws that we know work, and then we try to explain laws by doing something called theories, right? So basically what science is about is noticing the way that the universe is, right? And then coming up with explanations. So theory. A theory is an explanation for a law or a group of facts that has been confirmed and supported by a lot of evidence, and it can be modified or changed as new evidence is found. So a theory is basically explaining something that happens, that we know happens, that is true, but we're explaining it with tons of support. Like I'm talking about hundreds of years of evidence. It's not just something that's done once, or for a couple of years. It's like hundreds of years of evidence. Like the theory of evolution, right? We have tons of evidence for it over hundreds of years. But a theory can be changed, right? For example, we used to believe that the planets revolved around the Earth, and we had actual scientific data for this. But later on, we realized that we were wrong and that we're actually revolving around the sun. So it can be changed, right? And here, as an example, is the theory on cells, right? So if you look at the years, um, it went from 1595 to 1855. So this is hundreds of years, different people, different scientists working and adding to what we know about cells. So the first one in 1595, Jensen creates the first compound microscope, right? So then maybe like 100 years later, Robert Hooke, he's using the microscope and he's looking at cell walls. So now we know that cells have walls, but it took 100 years to add to that. Then in 1670, now we actually see living cells, things that are alive, and so forth. Now we're finding a nucleus. Okay. By 1855, we know that cells come from other living things. That's huge. This is a big discovery. So again, it's science. The way science is that we're all helping each other and adding to other people's theories to build on what we want to know about the world around us. Okay, and that's pretty much it. You guys have vocabulary on page 7. You need to fill out the vocabulary. And if you have this filled out and you're getting good grades above an 85 in class, then you can continue working on your research project. If not, then just keep staying tuned for more videos and keep paying attention in class and come in with questions if you're not really sure what to do in terms of this topic. So thanks for watching, and we're going to end this video.